Good morning, everyone, and welcome to Psychiatry Round. My name is Dr. Clemens Nyanjo, one of the Psychiatry chief resident here. Dr. Jeremiah is out. He's usually the one doing this on Friday. Today, we have a presentation from our third-year resident, Dr. Newt Agrawal. Prior to arriving here at ETSU for his psychiatry residency, Dr. Agrawal is a graduate of Massachusetts Institute of Technology for undergrad. He completed medical school at University of Oklahoma College of Medicine and St. James School of Medicine. He also received his PhD in biophysics, electrophysiology, neuroscience at McGill University. He has participated in research internship and other educational pursuits at Rush University, Department of Neurosurgery and Neurology, University of Chicago, Harvard Medical School, and even has work experience as a software engineer at Silicon Valley, California. He also has participated and been involved with several publication abstract and scientific oral and poster presentation. Here, welcome our true scholar, Dr. Agrawal.
There's a Velcro thing on her. Thank you, Clemens. Can everybody hear me? Okay. Hi, thank you for coming today. Today I'm going to be talking to you about the role of glutamate and dopamine and what ketamine has been teaching us about mood disorders. I got inspired to do this talk after going to a international clinical conference in Nova Scotia, which I helped organize this past September. And I heard two psychiatrists from Germany talk about ketamine and psychedelics. And they're interested in exploring basically how this medication works. And it got me thinking about antidepressants in general and my own understanding. And also, it reminded me of a talk that I'd heard by Paul Greengard, or I think I heard or read about, shortly after he won the Nobel Prize while I was a graduate student at McGill. And also, I had a visit with Ron Duman and John Crystal at Yale in the Department of Psychiatry, and that's when they informed me of the study, because they were principal investigators on this idea of using ketamine for antidepressant. So I did a lot of research, and I started thinking that there must be a common pathway for all antidepressants and mood stabilizers. And after doing some reading, I put this presentation together. I have no disclosures, but I am going to be talking about the use of an investigational drug, which I don't endorse. The medication or drug, if you will, is not FDA approved. It's investigational, and it can cause profound damage to neuronal circuits, even when used by professionally trained physicians or providers. I have some questions I just wanted to pose to everybody, if everyone get their clickers, and just try to answer these questions. I just want to see what you guys think as the answer. So that's the first question. Let's go ahead and poll. Everybody's done? Okay. Okay. So basically, after a major depressive episode, antidepressants work at which level? And there's a lot of different answers. Replenishing depleted neurotransmitter levels, upregulating translational proteins such as DARP32, increasing BDNF, increasing synaptic function, increasing synaptic density, increasing synaptic tree arborization, increasing the number of synaptic ligand receptors, modifying downstream messengers such as mTOR, ATK, ERK, GSK beta, and also possibly upregulating ion channels and synaptic density, or none of the above. Everybody ready? Okay, we're going to poll. So Okay, we got about 24 responses. I think I'm going to try to see what those look like. I've never used this before, so let's see. Maybe that's good. Okay, so the most common answer was B, which is upregulating DARP32. All right. And then the next is the first one. Okay, perfect. Let's go to the next question. Oops. Oh, what happened? To turn that off? Okay, so going to the next question. Okay, so if a patient was suicidal and had become treatment resistant, if ketamine or some other NMDA antagonist was approved, was available for use, okay, but it's not FDA approved, which of the following would you add next? So scopolamine, lithium, ketamine, an atypical antipsychotic, or ECT. Suppose we stop there. It may not work for this next one, but we'll try again. Okay, so I guess, I don't know. I'll go ahead and look at the answers here. So we got 4. All right, great. So let's look at what that looks like. Okay, answer B. Let's see. B is lithium. All right, very good. So we're going to move on, and we'll see whether or not everybody was correct or not correct. I'll get rid of that. Oops. Oh, great. Okay, where did my presentation go? I might need help. Okay, so the findings, actually, could you just make sure it's advancing? Oh, could you help me? Sorry. Good? No, it's not. It's not working from here. Is this working? All right, perfect. So the findings of the ketamine acting as a fast antidepressant is basically shaking the field of psychiatry, and it's led to the possible discovery of a whole new class of antidepressants. We're going to take a look at some detailed mechanisms of how antidepressants and mood stabilizers work, and possibly some new treatment strategies. So the story, so my purpose today is actually to provide greater clarification of how antidepressants work, and perhaps help everybody understand that there's perhaps even more that we can do for patients by combining medications and psychotherapy, and perhaps there is indeed going to be a new class of pharmaceuticals coming out of this work from the ketamine. So the story begins with Donald Hebb. In 1932, this Canadian psychologist postulated a theory that of learning, and the theory of learning involved synaptic plasticity. What he basically said was that when neurons are wired together, they fire together. So basically, if you have two neurons that are active at the same time, the synapses between them are strengthened, and thus the network between them is strengthened. This formed a theory of learning that was used to actually help justify the work of B.F. Skinner, and a lot of scientists got very interested in it because they were interested to find out if this might have been true, and it became the prevailing idea for about 50 years, and it still is a very common idea. So the work of Arvid Carlson, Eric Kandel, and Paul Greengard actually helped elucidate mechanisms of the Hebbian model for learning. And so Arvid Carlson is a physician scientist, and he discovered L-DOPA. He discovered that L-DOPA is actually not just the precursor to serotonin, but actually it's involved as a neurotransmitter. And what it does is actually regulate some basic functions within neurons. He actually is the one that helped promote L-DOPA's use for treatment of Parkinson's. Eric Kandel studied learning and memory in both the hippocampus and in a simpler model of neurons called aplesia. And he discovered that serotonin is involved in modulating learning and memory. He, together with Paul Greengard, helped further shape our understanding of long-term potentiation and short-term potentiation. Paul Greengard, working at Rockefeller University, discovered that when dopamine acts on neurons, what it does is it activates protein kinase A. And protein kinase A will activate cyclic AMP, which downstream affects a phosphoprotein called cyclic AMP. Actually, it's called dopamine and cyclic AMP protein phosphatase. Together, these three help formulate our understanding of learning and memory occurring in the hippocampus. Paul Greengard, I think, not only is a neuroscientist, but he's also got a PhD in computer science from Yale. I think he's probably one of the greatest scientists ever to have lived. This year, the Nobel Prize was awarded to John O'Keefe for work that he did while he was a graduate student at McGill, working in, under Ronald Melzack, who we know was involved in formulating pain pathways. John O'Keefe discovered CA3 placement cells. He also discovered that the encoding of the memories for these CA3 placement cells is regulated by harmonics of theta, basically in a process called theta precessing. He also discovered boundary cells within this area of the brain. Basically, these are cells that will help us tell distance to a border. This work is important because this region of the brain is involved in learning and memory. When I did my graduate work at McGill, I studied the entorhinal cortex, and the outputs of the entorhinal cortex actually, through the preferent path, go to the CA3 cells. And are involved in regulating a lot of our emotions and thoughts, and if they're perturbed, can cause epilepsy. About 10 years ago, there was a finding that occurred in a lab that won't be named that showed that an individual pyramidal cell within the medial entorhinal cortex can actually form a memory trace that's independent of network properties. And this was a Nature publication. And it was demonstrated in a lab I won't mention. So these two ideas form the foundation of what I'm going to be talking about today. So the stress diathesis model basically says that patients who have vulnerabilities to stress actually can have changes within their brain. And these changes can show up as downregulation of monoamines or upregulation of glutamate, or even changes in stress hormones or cytokines. So it turns out, depending on a person's vulnerabilities, they can actually go on to develop a mood disorder or even frank psychosis. The structural plasticity in the hippocampus involves synaptogenesis, neurogenesis, and dendritic remodeling, which ultimately converge. There's a lot of different biochemical pathways that influence this process. So what you guys answered earlier basically shows that you've actually learned very well the monoamine hypothesis of antidepressants. So this is the prevailing idea that neurotransmitter replenishment can somehow modify a patient's mood. However, there's other hypotheses out there, and one of them involves the glutamatergic model, and that basically means that whenever you have a kindling event, it results in downstream changes, perhaps even apoptosis and cell death. Well, I'm going to show that all of these pathways are actually linked, and it goes and the DARP32 protein, a phosphoprotein, is involved, and ultimately BDNF and a learning memory model is involved in how we actually develop mood disorders and treat them. So here we basically have two major systems, dopamine and glutamate. And these two major systems pretty much drive a lot of the neurons in our brain. And I'm going to show that ultimately they converge at this point here in regulating BDNF and learning and memory. And this is how the work that the 4 Nobel laureates that I just talked about comes in with importance, work we do. So the work that the guys who used ketamine did helped actually explore or even open up this kind of question. So we know that dopamine dysregulation involves the monoamine hypothesis model. We also know that glutamate dysregulation is sort of an output or a response to calcium-mediated excitotoxicity or apoptosis. So in the monoamine hypothesis of mood disorders, we know that a lot of these neurotransmitters, serotonin, dopamine, and norepinephrine, actually influence neuronal function. So we have psychostimulants, we have antidepressants, but they're all actually acting through monoamines, or are they? Then there's the dopamine hypothesis of psychosis, and we know that antipsychotics will block D2 receptors. And some people argue that they work, some people argue that they don't work. So both models of depression and psychosis are suggested to involve dopamine dysregulation, but I'm going to show you that both actually have dynamic interplay with glutamate dysregulation. So there's also, as I just mentioned, there's the glutamate hypothesis of psychosis, and basically this might be related to excitotoxic-mediated apoptosis or cell death. Other possible hypotheses, which I won't explore today, include the genetics of mood disorders, hormonal influences, as well as inflammation. But these don't really involve pharmacology, and we as psychiatrists don't have a way to actually treat these at this time, or I'm just not going to focus on it today. So I'll be oversimplifying some things. But that's just what it is. So ketamine. So ketamine is known as a rave drug. It's popular around the big city and some of the hippest clubs, and also, obviously, in the hippest of all festivals. The anesthesiologists use it frequently for inducing a dissociative anesthetic state because it's rapidly activating and rapidly off. Furthermore, now anesthesiologists are using it in ketamine clinics for treating mood disorders. Ketamine was developed in 1962. It's considered one of the listed WHO medications of essential drugs. It's basically a non-competitive NMDA receptor antagonist and influences a lot of different receptors, opioid, D2, it influences voltage-gated ion channels, and it can be taken insufflation or IV. Furthermore, it causes a dissociative state and can mimic sort of psychosis. It's because of the abuse potential, it's listed as a Schedule III drug. So again, ketamine clinics have started to pop up around the country. These are somewhat being run by anesthesiologists. Basically, the cost of the treatment is about $4,000, and each maintenance injection is $600 each. Basically, there's several infusions over a period of days. The efficacy ranges from therapeutic effects in mood lasting from 3 to 5 weeks to perhaps even 12 weeks. It tends to work better in younger patients. So Carlos Zarate in 2006 published the first study of the use of ketamine for treating treatment-resistant major depression. And here's his trial, and he basically selected 18 patients, and these are the results. So what he found after an injection, it was just a one-time injection. So basically, it was two days of injection, and they were separated by 1 week apart. So so patients actually ranged in age, but the average age was around 47. The youngest was a female. And she showed a 57% response rate. The oldest was a 60-year-old male who had also a 56% response rate. The mean response after a 1-dose IV injection of ketamine was about 56%. Overall, there's about 71% responders and 29% remitters. And even after 1 week, 35% of the patients responded. There was 1 dropout because of illness. And here you see after the first 80 or so minutes, actually after the first 40 minutes, you're already getting a response. And this response actually becomes sort of maximized about day 1, but maintains itself up to about a week. There's been other trials. This was done in 2002 by the same group at the NIMH. Basically, they looked at treatment-resistant bipolar depression, and they found that that in their study of approximately 27 or somewhere around there patients, that the response on the MADRS, the Montgomery-Åsberg Depression Rating Scale, was similar. Within the first 40 minutes, patients actually noted a response, and that this response maintained itself for up to 7 days. And this was done in comparison to a placebo control. So what if we had a patient who was suicidal, and you knew of a drug X, and you knew that it could prevent someone from committing suicide, but was currently experimental and not FDA approved? Would you prescribe it? Are you obligated to prescribe it? Is not prescribing it the same as failure to committing the patient? What options are there? Is there even an obligation? Well, it turns out this is a question that now we have to face. So in 2002, there was a study done, and it showed that ketamine rapidly reduces suicidal ideation after a single infusion in treatment-resistant major depressive disorder patients. Their findings showed that after 60 minutes of the infusion, patients reduced their suicidal ideation, and this maintained itself for up to about 240 minutes. So that's about 4 hours. So they used the Hamilton Depression Rating Scale, as well as the Beck Depression Inventory and the Montgomery-Åsberg. And in all those scales, suicidal ideation declined. Furthermore, there was a decline in anxiety. So could ketamine be something that could be used in the emergency room setting? Is it something that we might have to use? Furthermore, just this past year, there was another study that demonstrated that ketamine can reduce suicidal ideation. And this was in treatment-resistant patients. And they found that within 24 hours, they got an effect rating of approximately 0.58 compared to the midazolam control. So clearly, ketamine was having an influence. And just this past few months ago, they found out that ketamine reduces suicidal ideation in post-traumatic stress disorder patients within the first 24 hours. And you can see there's actually a doubling of an effect. So people were interested in trying to find out what ketamine is doing. And so what they did is they actually measured not just the response effect, but they compared the response to BDNF levels relative to midazolam controls. And they found that there's actually a correlation between an increase in BDNF levels with the use of ketamine, whereas the midazolam controls actually showed no effect, no correlation. So what's going on here? How are patients reporting such a dramatic effect after a single dose of a psychedelic? I mean, we've had monoamines for decades, and we've never seen an effect this strong before. One reason is that the antidepressants may be working on different receptors involving both the dopaminergic monoamine pathways and the glutamatergic, and that these might be interacting with each other. We know that dopamine is associated with several different psychiatric illnesses, ranging from depression to PTSD, addiction, ADHD, schizophrenia. Could dopamine signaling point the way to a final common pathway for how behavior is regulated and perhaps medications are working? Here's where DARP32 comes in. It's basically the master switch for neuronal signaling. This is the work of Paul Greengard, and he demonstrated that there's a variety of different receptors that actually all have inputs into the DARP32 system. So again, DARP is dopamine and cyclic AMP protein phosphatase. So we see that serotonin receptors, adenosine receptors, which are modulated by caffeine, opioid receptors, basically your D1, D2 receptors, NMDA, AMPA, GABA, and our ion channels are all having influences on this master switch. And we're going to get into this. There's going to be a lot of these figures, so no need to get too overwhelmed by this one diagram. So we know that from the work of Paul Greengard that DARP32 mediates the actions of multiple drugs of abuse. We know that it affects stimulants, hallucinogens, depressants, opioids. And we see here in this figure how these different drugs actually can influence neuronal signaling. And we see the effects of coffee, opiates, both delta and mu, and your basic D1, D2 pathways. And this is involved in the basal ganglia with movement and locomotion. We have our glutamate pathways and GABA, alcohol. And we see that it produces this, it modulates a phosphoprotein 1. And then there's a phosphoprotein 2. So these two are the products that actually are involved in intermediary signaling within the cell. And these also influence excitability. So again, the drugs of abuse, nicotine, ethanol, opioids, all are coming in to influence this signaling pathway. So here's the publication that Greengard did, the role of DARP32 in the actions of drug abuse. So here, if we look at the medial spinal neurons within the dorsal striatum, we see that caffeine actually affects the adenosine receptors and affects, and then we also see that Haldol can affect D2 receptors. What we find is that there's changes in the signaling for the DARP protein, and this occurs at a threonine residue, 34 or threonine 75, which is either upregulated or downregulated depending on the actual signal. We also see that cocaine influences D1 receptors and results in an increase in cyclic AMP and protein kinase A, also affecting DARP32. And as well, we also have the influence of amphetamines, both acting on the striaton Agrawal pathway as well as the Cummins pathway. So alcohol. Here we have alcohol basically causing a disinhibition phenomenon. And this is related to the fact that alcohol actually will influence NMDA receptors. So alcohol suppresses GABAergic inhibitory inputs to the dopamine neurons of the ventral tegmentum, and this results in increased dopamine to the nucleus accumbens, and this is interpreted as reward. And we see here, again, the alcohol will influence the DARP32 pathway. In fact, we noticed that, again, that there's transcriptional elements involved, and contrary to what people might think, over long term, the effects of DARP32 actually results in downregulation of CREB, which is involved in synaptoplasticity. Nicotine stimulates nicotine-dependent acetylcholine receptors, which also influences dopamine. And we see that here in this pathway, and this is basically probably the best figure I've seen so far. So what we have is nicotine will influence nicotine receptors, and this causes calcium signaling and downstream depolarization of the neuron. Basically, we have a direct path in which dopamine is being released through the snare complex. But here we have D2 receptors, which downregulate the effect of nicotine addiction. So we have the DARP32 protein pathway. Basically, there's two. It's a bimodal switch. So when threonine 34 is phosphorylated, you get a disinhibitory effect, and this results in more dopamine being released. When we get threonine 75 being stimulated, actually phosphorylated, we get the reverse. We get an inhibitory pathway. But this is the protein kinase A cyclic AMP pathway, which is involved in dopamine signaling within dopaminergic neurons, and is an important downstream pathway in which all drugs I will show work. Here we see the effects of cocaine on the D1 and D2 receptors within the basal ganglia, as well as caffeine. Both of them working on the same protein and causing motor changes. And then here LSD and PCP were found in mice to influence the same DARP protein. Here, mood stabilizers such as carbamazepine, valproic acid are acting at the receptor, but ultimately the downstream influence is affecting this signaling pathway. And ultimately, it goes down to affecting transcriptional factors and inducing synaptic plasticity. So we see drugs of abuse in that figure. We also see that antidepressants and atypical antipsychotics are working through mediating through this bistable switch. And I'll get into talking about GSK3 and mTOR in a little bit. So this is another diagram, and you can see right here in the center of it all is this phosphoprotein, which basically acts as an inhibitory switch. When you phosphorylate, then you get disinhibition, and then the other way, you actually get upstream regulation. And then the downstream pathways are modifications to genes such as cyclic AMP response element binding protein, and these actually have influences on protein function, synaptic vesicle, synaptic receptors, and synaptic plasticity. So here is another complex diagram, but you see the integration of all the different psychotropics, and we have here even cannabinoids, so marijuana will influence this pathway, and we have that occurring through calcium signaling, and then we have the influence of voltage-gated calcium channels, and then up here the NMDA pathway and the role of glutamate, and I'm going to get into this in a little bit. So what we have basically at the center of it all is DARP32. The reason I have so many figures is because no one's put all of this together in one diagram, and after doing a lot of reading, I actually kind of put this together. So basically, downstream mediation to the nucleus affecting genes, and then this is regulating proteins that are involved in synaptic plasticity. And so we also have DNA methylation being affected, and there's another mediator called mTOR, which I'll get into in a little bit. So basically, this is the basic signal. Dopamine acting through D1 affects protein kinase A, and DARP32, which then is involved in connecting to the glutamatergic system through NMDA receptors. And note that both types of calcium channels are involved. And we know that calcium signaling is really important for cell function and ionic channel excitability. Here we see the influence of DARP32, not just on sodium channels, on other ion channels, such as calcium. Paul Greengard showed in 2001 that Prozac actually worked on this pathway. And this is the publication that he showed that Prozac's influence on DARP32. So he demonstrated that SSRIs are actually working this way. And this happened over 10 years ago. So here we see the influence of norepinephrine, serotonin, glutamate, and we see how stress affects the entire pathway and how this is involved in upregulating or downregulating BDNF. Okay, and here is a diagram basically showing the connection between the dopamine system and the glutamate system. And again, in the middle is the DARP32 protein. So we see that this is a powerful mediator for cell function. And here again is where the phosphorylation sites occur. And so what basically this is if we look at engineering systems, this is basically a control system. So we have dopamine basically involved in rewarding behavior, and we have a feedback loop being driven by the glutamatergic system. And this glutamatergic system will basically either shut down this signaling pathway by and causing lots of calcium to flow in and triggering apoptosis and causing cells to die, or if it's stimulated in just the right way, it could actually cause a promotion of synaptic plasticity and, in a very, very handsome way, promote learning and what we're probably going to I'm going to tell you about here, the effects of ketamine. So So this is the control system idea I came up with in doing this research. Basically, dopamine acts to enhance behavior, and the glutamatergic system enhances to regulate that in a negative feedback loop. So this is like basic engineering. And schizophrenia occurs when this is overactivated, because you have cells getting rearranged and learning being occurring in an abnormal fashion because circuits are messed up. Okay, the work of Ongor and Yüksel showed that the glutamate receptors are actually affected in mood disorders, and this was brain imaging work. Furthermore, we know that calcium-mediated excitotoxicity is involved in apoptosis, and we know that it's also involved in fear extinction and learning and memory, and that we know that the glutamate system is involved in psychosis and dissociated feelings. Glutamate is actually a neurotransmitter that affects a couple of different receptors. They're the NMDA receptors, the kainate receptor, and simple glutamate receptors. There's a whole family of these. Basically, how it works is this is both a voltage-gated and a ligand-gated ion channel. This is absolutely fascinating if you think about it. That means, because most of the ion channels that we have are either one or the other, voltage or ligand-gated. But this can be both. So what that means is that NMDA receptors offer a powerful way of regulating learning, because of this very property, it allows the NMDA receptor to be serve as a sensitive switch, so not only responsive to excitability, but also to chemical pathways. We know that the brain is regulated by both chemicals and electricity. So when we have an activity that we want to reinforce, if two cells fire together, you get that response through voltage-gated activity, and this triggers a cascade which enhances synaptic plasticity. Furthermore, you can have chemical stimulation through something like dopamine, through GABA, and that's where the ligand-gated element comes in. Nature created a wonderful receptor, and that's the NMDA receptor, and that's where ketamine is actually exposing the wonderful properties of receptors. So it turns out the way this works is that kainate receptors actually have to be activated to turn on the NMDA receptors, and I'm going to get into that in here in a second. So this is a model of the NMDA receptor, and we notice that there's several different subunits, NR1, NR2. In order for the NMDA receptor channel to open and mediate calcium influx, you must have serine and glutamate. These are coagonists. They must attach. Furthermore, there must be a depolarization of the cell membrane that allows calcium signaling to occur. And this we know is very important, because if we get too much calcium, we get cell death, apoptosis. And a few weeks ago, we had a Grand Rounds talk demonstrating what the effects of that is on the genes, as well as like telomeres. We have other ways of actually regulating this, and magnesium will actually block this channel. And there's sites for ketamine. The ketamine site is possibly the same site as the PCP site, but I haven't found any papers actually showing that to be true. This ion channel is a multi-membrane, four transmembrane protein, and there's work right now being done to develop new antidepressants, new antipsychotics that actually work on not just the subunits, but possibly even agonist sites. There's work being done to develop a glycine-like agonist. So the effect of doing this is, instead of having ketamine-like influences, because we're blocking this part, this is what causes the psychomimetic effects, the hallucinogenic effects. Drug companies are working on modulatory pathways through coagonists, through affecting NR1 or NR2. NR1 is absolutely necessary for this channel to function, and there's the expression of the other subunits, NR2. So anyway, so why does glutamate activation and NMDA receptors not cause excitotoxic cell-mediated death? I mean, after all, we're dealing with calcium signaling. Well, it turns out the answer to this is involved in the fact that NMDA receptors can be both intrasynaptic and extrasynaptic. And this is a very, very important finding, because these extrasynaptic NMDA receptors that cause cell death. And so whenever these get overactivated, you get downstream apoptosis. But we get nice, beautiful signaling if we're stay within within activating the NMDA receptors, and that goes through the signaling cascade that I've been talking about. Here's an example, a figure demonstrating how NMDA receptors work. Basically, what's very important here is the role of glia. We've always wondered, are glial cells involved in regulating synaptic transmission? Are they involved in regulating mood? There's been some recent studies that have shown that when someone's depressed, it's not just a local phenomenon. The entire brain is affected. The white matter tracts are affected. There's actually changes in the whole volume of the brain. So a paper was just recently published, and it showed that if you give psychotherapy, actually cause a remodeling of both the white matter tracts and the gray matter tracts, and these actually come back up. So when you combine the therapy with drugs, you actually get this upregulation. Anyways, the point here is that we have glutamate signaling occurring through glia, and how this works is basically what's going on with ketamine is it's blocking the interneuron NMDA receptor. And I think I have another figure that I'll talk about, and I'll show you how that happens. But basically, AMPA has to actually has to open, and then it sends a depolarization signal, which then opens the NMDA receptor. Here is another figure showing the influence of NMDA on CREB, and this ultimately has downstream pathways that are important for nerve cell regrowth, or actually remodeling, and synaptic plasticity. And you have recycling pathways of glutamate involved with glutamine, and we see the important role of astrocytes. That's why the overall brain health is very important for synaptic transmission. And here is a diagram showing the role of both the extrasynaptic NMDA receptors and the intrasynaptic. So basically, we overstimulate these extrasynaptic, we cause apoptosis, things such as like Alzheimer's, neurodegeneration. But if we stay within the cell, we can actually regulate our function better. So where is this convergence occurring? Well, it turns out BDNF turns out to be a very important little protein. So it turns out BDNF levels are increased in response to exercise. They're also increased in response to seizures. They've been shown to be increased in response to antidepressants. In fact, BDNF itself is an antidepressant if you take it directly. But it turns out in certain populations of neurons it actually causes depression, such as the hippocampus. Furthermore, BDNF prevents excitotoxicity. And what's surprising is it actually increases in response to lithium. I just talked to you guys about how BDNF affects the hippocampus. We also know that serotonin is involved in interacting with BDNF, and that it's involved in the role in affecting mood. So, furthermore, if there's depleted levels of BDNF, it leads to obesity and hyperactivity. Furthermore, if there's genetic polymorphisms, it can result in suicidal behavior. It can result in changes in mood function. Furthermore, it's been associated with passive avoidance in mice within the hippocampus and entorhinal cortex. So the interface looks like it's occurring through these mediators, DARP32, mTOR or GSK3, MEK, and other pathways. And the convergence for all of these little signals appears to be BDNF, CREB, and CFOS and CJUN. So what's happening is there's changes in synaptic plasticity, and this can be seen as changes in function, which involve timing, frequency, amplification, dynamics, cluster expression, speed. These are concepts that you don't normally see in the literature. I wrote these in because that's what's happening. These are engineering type of effects on burst firing and stuff. And also you get changes in morphology. You get changes in density, arborization, volume, distance between signals, and orientation and interconnectivity. And if it goes bad, you get apoptosis. So there's this interplay between apoptosis and neuroprotection. So we know that different forms of antidepressant treatments can affect structure, and we see that SSRIs actually affect the anterior cingulate gyrus and area 25, and the role of TMS, ECT, DBS, and simply memory. We can actually learn depression, and we can remember it, and that can actually promote So what we see is when we give SSRIs, there's a down signal of this area, and if we remember a bad event, we have an upregulation or overactivation. So we note that there's actually a atrophy of an area of the brain in postmortem brains. Actually, the glia is actually decreasing in size in response to stress. So, so what do antidepressants do? Antidepressants are associated with neural plasticity, basically synaptic remodeling and synaptic contacts, as well as increasing synaptic strength. We noticed that because there was a study by this group here, by Bessa and others, basically showing that these antidepressants reverse the effects of morphological changes. So here we see a normal brain. These are normal synaptic connections. In response to strong amounts of stress, we actually have a paring down of synaptic vesicles, and there's actually a decrease in branching, a decrease in synaptic connections. So what's happening is everything we've learned, the Hebbian hypothesis, is getting downregulated. So happiness is being downregulated in response to stress. You could argue, I just came up with that ridiculous idea. But then we give, if we give an antidepressant, we notice that there's actually a reverse effect, and we get upregulation of synaptic boutons and more arborization, and we get a healthy-looking dendritic tree. So it turns out all of our antidepressants are working by this basic mechanism. And this is my overall learning point for everybody today, is that when we're giving medications, they're really important. And the reason why they're important is they help restore brain anatomy. So when someone has a mood disorder or when someone has psychosis, it is not in the mind. It's not just in their head. There's actually changes in structure. There's changes in synaptic function. And when patients don't take their medicines, they do not replenish their connections, and they do not get better. And we see that clinically, but we have to send that message that this is a real disease. These are real diseases, and there are structural changes in the brain. This is the effect of ketamine on the synapses of mice. So they found, this is their control mouse. It's a chronic stress mouse, and you see there's a downregulation of synaptic boutons and spines, but within minutes, within like 40 minutes to an hour, there is this dramatic effect of upregulation of synaptic spines. So we see the powerful nature of ketamine when given at low dosages can actually reverse the effects of chronic stress. And here we see the influence of ketamine on different pathways. And here there's an influence on protein kinase A, protein kinase C, CREB, as well as upregulation of BDNF. So ketamine ushers a new wave of antidepressants called rapid-acting antidepressants. And the reason this happens is it causes a burst of glutamate to increase synaptogenesis, just like LTP. Basically, ketamine is helping us get a greater understanding of the interplay between dopamine and glutamate systems. And it may offer an ability to actually, if we're smart about it, regulate mood in patients. So here is the actual event that's occurring. So we have regular mood and normal flow of like of glutamate influencing AMPA receptors and NMDA receptors, and this is a healthy little neuron there. But when someone gets exposed to stress, there's a downregulation of BDNF. mTOR pathways are affected, and we get a long-term depression-like event. And this causes changes. It turns out this is the DART protein pathway, and phosphoprotein 1 will increase GSK3. Well, GSK3 will promote depression, and we further have signaling from the GABA interneurons, and this little NMDA receptor is really important. So what happens is when we give ketamine, we get a glutamate-like burst. Because what we do is we're blocking this NMDA receptor on the interneuron, on the GABAergic cell, which is putting the brakes on glutamate release. So ketamine blocks this. blocking this, it causes a burst of glutamate into the synaptic vesicles, synaptic cleft. And this increase in glutamate will influence AMPA receptors, which then send sodium into the cell. That causes depolarization. Then that causes depolarization of the NMDA receptor, and only if glycine and or serine and glutamate are attached as coagonists, and does the magnesium block go away, and we get calcium regulation into the cell. So it turns out that that is involved in upregulating BDNF and promoting the effect of antidepressants. We also get an increase in spine synapse number and function. I want to emphasize function. And this is, again, the pathway. One thing that we have recently learned is that lithium actually influences GSK3. Well, people have always wondered whether or not lithium influenced what it was actually doing. There's thoughts of it affecting the inocetyl pathway, but it turns out it also influences GSK3. Well, GSK3 is a very important protein intermediary because it is involved in apoptosis and inflammation. So the reason why lithium works is because it helps to stabilize that. So ketamine is basically telling us that there's actually biological memories that can be unlearned or relearned. And that might be the effect of the antidepressant. So it's not because of a dissociative or hallucinatory role that ketamine is doing. It's not getting high. And I want to emphasize that because the average layperson would think that you're taking dissociative psychedelic and that's why you're feeling better, because you're dissociating away from your troubles. But that is not what's going on. So again, psychiatric illness is not in all in our head. There's actually invisible functional changes within the brain. There's also, as well, there's structural changes. And NMDA receptor antagonists can be extremely dangerous if they're misused at the wrong potency levels. There appears to be a final common pathway, and that this common final pathway is emerged at where we get this idea of neuroplasticity, and that's where learning and psychotherapy is very important in therapy. So basically, medications can be used to stabilize a patient, and psychotherapy, the reason it's important is it helps with slower long-term potentiation. Medications help with both short-term potentiation and long-term potentiation. It helps in stabilization. It helps with improving structural changes. But the greatest amount of benefit that we're going to get is from both psychotherapy and medications when given together. And we know that from all of our patient studies. So ketamine is believed to upregulate BDNF levels. It appears to be very critical in the role of synaptogenesis, and it also influences the prefrontal cortex. Here's mTOR. mTOR is what Dr. Duman's group at Yale believes as how and why ketamine acts so fast. He believes that it's actually taking a shortcut. And so mTOR is a part of this whole intermediary pathway, but this allows more rapid changes in cytoskeletal and synaptic density and synaptic receptor changes at the dendrites, and that might be the reason why ketamine works a little bit faster than what you would think otherwise. So, and this was the publication by Dr. Duman's group, basically that mTOR-dependent synapse formation underlines rapid antidepressant effects of NMDA. There's another very important kinase, and it's called glycogen synthase kinase. It's a serine-threonine kinase. It's involved in phosphorylating a serine or a threonine residue, and it's very important because it has been discovered to be involved in controlling inflammation, and it's been involved in controlling mood, Alzheimer's disease, diabetes, and cancer because it's involved in energy metabolism, cell proliferation, apoptosis. It's actually found in regulating tau within neurofibrillary tangles. And when it's inhibited, it can suppress neuroinflammation because this in itself may be a mechanism for causing changes in mood and psychosis. And we know that because there's a whole schizophrenia model of psychosis that suggests that maybe a virus did it. Anyway, so we know that GSK3, when it's inhibited, will suppress interleukins and chemokines and tumor necrosis factor. So So it turns out lithium acts on this. So lithium inhibits GSK3, and that feedback loop is regulated by DARP32. So this promotes cell survival, synaptic activity, cellular plasticity. It also promotes BDNF and synaptogenesis, neuronal survival. And everyone who's ever done patch clamp recording knows how amazing lithium is when you're recording. So, so basically, we're expecting a new class of antidepressants. There might be AMPA receptors or NMDA receptor antagonists. AMPA receptor agonists or potentiators. So basically, blocking NMDA receptors is thought to increase glutamate within the synaptic cleft, which activates AMPA receptors, triggering a voltage depolarization that activates NMDA receptors on the postsynaptic membrane. Together, this release a cascade that ultimately results in release of more BDNF, and that promotes antidepressant effects. So drugs are being developed, basically, possibly to act on AMPA, basically to serve as a potentiator. The reason why AMPA is so important is because it doesn't cause the psychomimetic effect. It doesn't cause the hallucinatory effects. It's sending a depolarization signal to NMDA. So it's a downstream. It's a control on the entire thing, which can shut and kill cells. And that's NMDA-mediated calcium release excitotoxicity. So AMPA is a great target for drugs because there's different ways to actually very carefully regulate the effects of NMDA. So there's other targets that include the subunits on the NMDA receptor. There's competitive targets that people are investigating. We know that memantine is being used to treat Alzheimer's. It's a non-competitive antagonist. It turns out that competitive antagonists are what are associated with the psychosis, the psychomimetic effects, so they're trying to stay away from that. Other kinds of drugs that might be developed are voltage-gated ion channel modulators, agonists and antagonists. We know that these currents are really important in regulating mood. I studied the persistent sodium current. Basically, these currents are involved in shaping oscillations on cell membranes, and these electrical oscillations, if they're properly controlled, can cause excitability, and that can trigger your NMDA-mediated cell death or changes in mood, changes in attention deficit, or psychosis. Basically, there's also this hyperpolarizing cationic current. The interplay between that and that is involved in oscillations and breathing, respirations, basically sleep patterns, virtually the entire brain. So these are other targets. And then there's the NMDA receptors. There's already drugs being developed for glycine. There's D1. There's kainate possible targets. There's downstream protein kinases and phosphatases. There's mTOR and then MEK. Furthermore, people might be developing transcription factor regulators. Furthermore, late gene products such as BDNF. And then there's the epigenetics, which drug companies might be targeting, where there's either increased cell transferase or DNA demethylation, or down gene silencing through DNA methyltransferase. Basically, this is my warning to the world. If you give the key to launch the nation's nuclear stockpile to an absolute moron, you certainly will get to bring the Berlin Wall down, but probably not the way the world was hoping. NMDA receptors offer an ability to fight depression. They actually might be very, very influential in not only decreasing suicidal ideation, but stabilizing depression very quickly. But if they're used in the wrong hands, might lead to a tremendous amount of damage. These are the drugs that are under development. Currently, there's this many number of clinical trials. And they just published the results of, actually they just completed the glycine 13 study. It's a glycine antagonist. It just finished phase 2. There's 80 patients in it. And we don't know the results, but if we look at this table, they're developing NMDA receptor antagonists. There's a whole bunch here, and they're looking at safety. And then there's a traxoprodyl, which is an NR2B. That's an NMDA or glutamatergic subunit receptor. and then we have glycine and other pathways. So in conclusion, synaptic plasticity and remodeling appears to be a key process for the therapeutic efficacy of treatments for patients from mood disorders, which at the cellular level are reinforced from environmental or epigenetic influences. When treating patients, medication management paired with regular psychotherapy treatment is proven to be more effective than medications alone. Long-term potentiation happens not just at the network level, but also at the individual single neuron level. When the patient is stuck and becomes treatment resistant, we might need to consider more than one strategy. Yeah, I'm ready for questions. Thank you for, sorry.
This is not working, so we need to just speak up if you have a question.
I wonder if you could tell me something about S-ketamine. That's still something that's being looked at, non-psychoactive.
Oh, I wonder if it's like, I actually don't know anything about it, but I'm wondering if it's acting in a slightly different way, like at a different site. Yeah. So that's what the goal is to basically not block the inner pore of the channel and maybe look at downstream ways of affecting that the NMDA receptor. So that's where the GLX13 medications coming in, the AMPA receptors, the AMPA potentiators, NR2A. So and the coagonist sites. So yeah. So it turns out that in the stream of NMDA receptor antagonists, the most potent is MK801, and then the next is PCP, and then after that is ketamine. So ketamine is not your worst nightmare, but it does raise the question about bath salts, and I do believe bath salts are affecting this pathway, and that's what's causing the long-term changes that patients see when they take those drugs. Yeah, go ahead, Dr. Miller. Okay, that's a very good question. The question was, what is the duration of the benefit from ketamine? So the studies so far are seeing the effects last as long as up to about 1 to 2 weeks. There's been some anecdotal reports in some people where it's lasted longer, up to 4 weeks, 6 months. I think Dr. Crystal told me 6 months, and I was like, anyways, so the idea is that people would come in for regular maintenance infusions, but this is the first time, I believe, and I'm still new to this field, I would say, that we've ever seen an antidepressant so powerful that can cause such a strong response, so that you give one dose, and that dose effect lasts as long as 1 week. So the MADRS and the Hamilton rating scales are still at the same level a week later. Yes, Dr. Griffith.
What concerns are there about maintenance ketamine therapy?
So that's a very good question. So the question is, what concerns are there about maintenance ketamine? There's a lot of concern, and it has to do with the nature of NMDA-mediated excitotoxic cell death. We know that cognitive function can be impaired, from things like PCP and bath salts, and if people misuse it, it can cause long-term changes. But one of the concerns is we don't know what the long-term impact is from administration of ketamine. They believe that it's not going to be a medication that's going to be used for treatment, and so that's why there's such a huge interest by the pharmaceutical companies to develop less potent activators of the same pathway. And so that's where those subunits are coming in. That's where the AMPA potentiators are coming in. And the one thing that I'm wondering about is why people aren't targeting DARP32, which is really kind of like the master switch. So even Duman's group, that publication was just on mTOR, but I hadn't seen any literature making that connection. But we know it's there, so that's what I'm really wondering about. Anyways, yes. Okay, so there's actually two questions in this question. So the question is basically, if I was to summarize, how is ketamine going to come into play relative to say something like ECT in terms of like I guess maintenance? So and then but the other part of that question is comes out of that, which they've actually shown, is that when ketamine is used together with ECT, there's actually huge improvement. Okay, but so the first question about ketamine maintenance, I don't know. That's really the big question, is what does this look like in the longer term? We know acutely that it has a clear effect. And we don't know how cognitive function is being affected. We don't know what other changes are occurring and how long this might have to go, or even if there will be downregulation. But what's really fascinating is what we're learning about the pathway. And I think that's where there's going to be other medications that are going to just take advantage of it. As far as the fact that they've combined ECT and ketamine together, that in itself might be the ideal combination. So we're going to get to see that later. Dr. Moore.
I was at the same presentation mentioned what impressed me was these people who were doing these studies, healthy control, they had tried ketamine and they had quite severe, troubling psychotic side effects. It's not ever going to be a treatment that we would want to give to popular patients, but as you say, it's a great way to learn more about how depression affects the brain and how we might probably give medication.
Right, so Dr. Moore had mentioned that he was at the same presentation in Nova Scotia as I was at, and these two German scientists, psychiatrists, they had actually taken the ketamine themselves while they were doing the study with the patients. And so they reported the psychomimetic, psychedelic effects, and it just raises that whole question how we do experiments and how they do them. But anyways, so that got that was where kind of it got into this. The study was the comparison with midazolam. Midazolam is an anesthetic. It's not even a NMDA receptor antagonist. So that's what got me thinking is like, well, why'd they use midazolam? Why didn't they use PCP as a control or something else? Anyways, so that's where I started thinking about there's clearly a whole different pathway in how antidepressants are working. So I started looking at this. But yeah, we're not going to use ketamine as an antidepressant. It's going to lead to other drugs. And I think it's just a putative model. I mean, I appreciate the work of Dr. Zarate, but I'm not sure if that's going to be the final drug. Yes, go ahead.
I just want to mention that medically, the problem that ketamine users present with is bladder acid.
Yes, good point.
It's extremely
Mm-hmm.
and the bladder has to be replaced. So until we solve that problem, it's not going to be
Mm-hmm. So what was said was that ketamine causes bladder toxicity, and the damage is severe. That's true. It also causes liver toxicity, and you know, it can clearly wipe out your memory centers. So you can watch the YouTube K-hole videos. I was watching those last night. The problem with ketamine and the whole story, this whole idea of that's been now so well talked about around the country and all of the psychiatric presses, is that kids are going to think, oh, well, if ketamine cures depression, I'm just going to go get it off the streets or at these festivals. And, oh, if a small amount isn't good enough, I'm going to use a larger amount. But that's exactly where the problem is. Ketamine actually has its influence in this manner at very, very low dosages, but kids aren't going to understand that. And when you take it in a high concentration, that's where you're going to trigger the calcium-mediated excitotoxicity and cell death. Yes?
Just wanted to mention, if you haven't mentioned it, that you can get this later effect of the blunting of the emotionality receptor.
Right. That's another NMDA receptor antagonist, and it turns out dextromethorphan is an effective antidepressant. I have one patient at the VA who's abusing it, and it's been hard for everyone who's seen him to get him off that. And apparently, he doesn't talk about it, but it must be causing him some sort of antidepressant effect. But I was able to get him switched over to Wellbutrin for now. But yeah, so dextromethorphan might lead to some interesting, if people investigate how that's working, some interesting medications. Yes?
Following up on Norman's comment about adverse effects and normal control, what happens when ketamine is usually used to treat chronic pain? Sort of a psychiatric complication to that process?
Right, so ketamine can be used for pain relief, and that opens up another whole range of questions because we know the complexity of pain. I actually don't know the answer to that. So, but that offers another interesting question, and who is going to, if this line of medications actually comes online, who is going to be administering this? We already have anesthesiologists who are making the argument that, well, we've been using this drug for 30 years. We know how it works. We can do it safely. We're anesthesiologists. It's given IV. So they begin to open up these clinics, and they're treating psychiatric patients. I mean, that in itself is a huge question. Who's going to actually administer this? And then are the pharmaceutical companies going to come up with a different route of administration? We do know that they're working on insufflation, and then I guess sublingual. So what may happen is that they'll use it in emergency rooms. So you get the acutely suicidal patient, and maybe it could become a medication to stabilize people before they go to an inpatient or anywhere else. Yes?
I just had a question about the epidemiology of this. Probably, I think, the population that most commonly administers IV ketamine, pediatric oral children. Do those children have any sort of different affective prognosis?
Yes, these are really good questions. So you would think that people would actually have looked at all this before. Yeah, that's a really good question because that offers possible answers to the long-term changes or plasticity of the effects of ketamine from early childhood onward. So I haven't seen that literature, but it's a very good question.
I don't think the pediatric oral children follow that.
I think the surgeons and the anesthesiologists think that ketamine is pretty benign. Yeah, in short term. Yes?
We didn't get a chance to talk about specificity of cytokine effects, and I was just wondering if you had something to say about how pro-inflammatory cytokines like TNF-alpha, IL-1, and IL-6 are downregulated during excessive response in IL-10 receptors related to infectious dosing.
Oh, yeah. So I don't really know the details of that interplay, but yes, this is part of the other story is neuroinflammation. And I think when we're treating depression, that's the one thing we don't really go for. We don't really go after. But I do believe there's probably an inflammatory response that's occurring that's affecting people's mood and contributing to things such as like frank psychosis. But it turns out ketamine does influence the cytokines. And I don't know the specific ones, though, but that's the other equation, or one of the other equations.
And I also wanted to
Yeah, so what's interesting is these pathways that I presented to you help me at least, understand why valproate, lithium, and carbamazepine are our most effective mood stabilizers, because it shows the downstream pathways. So GSK3, for instance, lithium, and I guess valproic acid. So I'm sure that's what they share. And the potency levels, why I'd say Depakote doesn't work as well as, say, lithium, probably has to do with what's going on as intermediary in the signaling. And so that's why lithium, for instance, is so wonderful. But lithium has other, actually, it's not just lithium. I think it's, there's some talk about inositol. So it's interesting questions. Yes? Oh, okay. Yeah, so those are questions that they're working on. Like these studies are so fresh, and there's so much material I looked at, because I'm looking at, this is not my area. So I was looking at literature that I had never seen in my life. Anyway, so there's so much stuff coming out of this at such a rapid rate that it's become almost, in a way, the end thing. Especially with funding for projects, there's more NMDA-related research funding right now than, say, on GABA or something. But yeah, activation. These things could be something that maybe side effects or upstream effects that we are not really aware of or thinking about. Thank you so much.
I think we should kind of stop at this point. Thank you.
All right.